Welcome back to another breakdown. Hell yeah, I'm excited because we're doing a BB passage breakdown. Okay, BB passages, they are my favorite because it's kind of like the closest thing that you're going to be learning in medical school. So this stuff is cool, guys. I have fun doing BB passages. Yes, I said that. I have fun doing BB passages. As hard as that, as much as you want to believe that, I have fun doing these passages. Okay, so we're going to do a passage here that I haven't done before. Okay, I'm just gonna, this one I did, this one I did, this one I'm pretty sure we did a lot of these. I think passage 8 we didn't do. Yeah, passage 8 we did not do. Okay, so I'm gonna show you guys exactly how to read this, the pace to read this, how to pick the best answer choice. I'm gonna show you guys how to understand these figures here. Okay, I'm glad that we have somewhat complicated figures that I can help you guys with. All right, I'm going to show you how to pick the best answer choice, what's a good answer, and what's the best answer. I'm going to show you guys how to get in that AMC MCAT logic, okay? Once you understand how the AMC, once you get inside the test maker's head, guys, this becomes a piece of cake, literally, okay? Just get in that test maker's head. That's it, all right? No one, I say this all the time, okay, but no one goes through undergrad doing MCAT passages, all right? This is a skill set you have to develop, all right? So, as always, guys, let's get into it. No more talking. All right. What you're going to do is read the passage first. Okay. I'm going to scroll through this here. So I'm scrolling through. Okay. I want you guys to read the passage and then write down your answers for these questions. Okay. Write them down on a separate sheet of paper. And then I'll explain why you got things right, why you got them wrong. Okay. So pause it whenever you need to, guys. This is question 1. All right. This is question 2 here. Second question, third question, fourth question, fifth question, and the sixth question here. I'll scroll down a little bit for you guys. Boom. There it is. Okay. Do it first. Make sure you do it first, guys. All right. Crack my neck. Crack my fingers here. Get ready because this is going to blow your mind. I promise you. Here we go. All right. The retinoblastoma protein, PRB, has a primary role as a transcriptional co-regulator of genes involved in several important cellular processes such as the cell cycle. Boom. First sentence right there. We got a lot going on here, okay? You guys have to understand, prefixes, suffixes, all that for the MCAT, okay? Whenever you see some oma, blastoma, carcinoma, that means cancer. Okay, oma means cancer. Retino, that means in the retina around there. So retinoblastoma protein, it's a protein, it's involved in cancer of the retina. Okay, and they tell you right here it's involved in the cell cycle here. Okay, what does it do? In my head, I'm already thinking, does this protein, does it help the cell cycle? Does it hurt the cell cycle? Does it help cancer? Does it hurt cancer? That's already going on in my head, just a little bit, okay? But let's keep reading. All right. Researchers hypothesized that PRB may also play a role in mitochondrial apoptosis. Boom. What type of apoptosis? Mitochondrial apoptosis. All right. The mitochondrial apoptosis pathway involves the activation of the BAX and BAC proteins, which results in the release of the caspase activator cytochrome C. Cool. All right. Fortunately for some of us, okay, not all pre-meds, but we kind of went over the BAX and BAC, apoptosis, mitochondrial pathway in our undergrad. In cell and molecular biology, I had a class, we went over this. I don't know if you guys have, but for me, that's unfortunate that I did. But we're going to keep going here. This is all making sense here. It's all making sense, all right? Let's keep going. And remember, guys, the pace that I go through this is the pace that you should be going through your MCAT. This is the proper pacing here, all right? The way I read it, the pace that I read it. To investigate a potential role for PRB in this process, researchers used stable cell lines of wild type, BAC deficient, and BAX deficient rat embryonic fibroblasts in which PRB expression could be induced. PRB induced above basal levels, PRB basal. Okay. So we got a lot going on here again, guys. Do not get lost in the details, all right? It's not too bad. Honestly, not too bad, all right? Again, some suffixes and prefixes you need to know. Whenever you see blast, okay, fibroblast, cytoblast, that means stem cell, okay, stem cell. So what are they doing here? They got these cells, All right, they have the wild type to compare. They have one that's deficient in BAC. They have another one that's deficient in BACs. All right, they induce PRB in some of them, and then they just leave the PRB in their basal levels. And they're seeing, hey, what's the difference here? What do we got going on here? Does apoptosis increase or decrease? That's it, okay? Cells with basal or induced PRR expression for 24 hours were treated with the pro-apoptotic factor TNF alpha for 48 hours. Boom, I'm gonna highlight this because it's very important for you guys to understand that it's pro-apoptotic. Pro. We're doing apoptosis. When we have this, it's going to make apoptosis happen. I'm highlighting it just so I can have a little, reminder in my head. Hey, TNF, pro-apoptosis. TNF, pro-apoptosis. Okay. Then, the percentage of apoptotic cells in each group was determined by staining cytoplasmic phosphatidylserine with the fluorescent marker annexin V. Okay, cool. Here's a figure. We do not look at the figure. Not yet. When the question asks, we look at the figure. We don't want to waste time analyzing this, okay? So you're going to go ahead and skip the figure. Okay, boom. Constructs were generated to contain inducible versions of the 3 domains of PRB, including the N-terminus, all right, residues 137.2, the small pocket domain, residues, and the C-terminus, RB, C residues, cells with basal levels of PRB, inducible PRB, or the 3 variants, RBN, RBSP, and RBC, were treated with TNF alpha for 24 hours. Then, the percentage of apoptosis was determined by NNXV staining. Okay, cool. Very easy, guys. I have a great understanding of this passage. Great understanding, okay? What do we got going on here? Pause the video right now. Try to say a summary of this passage, okay? Say a summary about it. All right, cool. Got your summary? This is what I think. All right, I think that these researchers are interested in studying apoptosis. What does this retinoblastoma protein do? They're proving the fact that PRB has an important role directly in apoptosis. That's what they're proving, okay? Does PRB interact with BACs and BAC? Yes, it does. What happens when it interacts with BACs and BAC? Well, when it interacts with BAC, we have induction of apoptosis. When it interacts with BACs, maybe we have inhibition of apoptosis. We don't know until we look at the figures, okay? So that's what happened here. And then here, they just made constructs. There's not much going on here, okay? Now, some pre-meds, they ask me, hey, Eric, when do you really pay attention? When do you really dial in the passage? When do you kind of just read through it, to save time? And whenever I see here, 137 to 2, 37 to 376, that I just run through it. I don't really care. Okay, I don't care the specific numbers, this residue to this residue, okay? I only care when the question asks for it. The question will make it known that they're referring to a specific residue, okay? So over here, the numbers here, just briefly go through it. All right, enough talking, let's get into the question here. In order to determine the effect of PRB on tumor growth, tumor cells containing no PRB, basal levels of PRB, or additional induced amounts of PRB were injected into mice. Tumor cell growth was measured at 11 days post-graft. Which graphic shows the expected result of this experiment? All right. As always, guys, whenever you read the question, I always recommend to hang on to every word. Yes, hang on to every word in that question. You want to understand that question in its entirety, okay? So they're asking me, okay, what's this volume? What's the tumor volume when we have PRB, PRB induced, and we have no PRB? PRB knockout. Remember that minus means knockout, okay? So in order to understand this, guys, we have to look at this figure here, all right? We have to look at this figure. When we induce PRB, does apoptosis go up or does apoptosis go down? All right, let's look at the figure here. When we induce PRB, black is PRB basal, gray is PRB induced. Let's look at the wild type. Always look at the wild type first, guys. It'll make the whole entire figure way easier, okay? Look at the wild type first. Completely wild type. All right, so we look at TNF alpha when there's no presence of TNF alpha. Here's the basal levels, regular PRB in the cell. When we induce it, we have more apoptosis. Let's look at the wild type again here with TNF alpha, regular. We have good amount of apoptosis, but when we induce it, whoa, look at that. Huge apoptosis, okay? So my conclusion is that PRB, when induced, increases apoptosis. Very similar, very easy. Very, very easy. Okay. So I'm looking for that. When we induce PRB, we should have high apoptosis. But they try to trick you here. MCAT logic 101. Okay. Apoptosis, volume, tumor, they're always going to test that. Okay. When you have increased apoptosis, you have a decrease in tumor volume. That's the connection you have to make. That's the logic that AMC wanted me to make in my head. That is it. And now that I know that, this question will be easy for me. Okay. So when we have induction of PRB, tumor volume goes down. So let's see over here. Let's check A. Here we have PRB basal. When we induce PRB, we have an increase in tumor volume. If we have an increase in tumor volume, that means apoptosis went down. That conclusion here in A is wrong. We should see a lower bar for PRB induced. Okay, because PRB increases apoptosis. Bam. That's wrong. Here, again, we have the same thing here. PRB induced is higher than PRB basal. That is wrong. Okay, PRB induced should lower the tumor volume. Bam. Next one here. Good. PRB is lower than PRB basal. Good. Okay, let's check this one here. This is saying PRB induced and the RB knockout, okay, is relatively the same. Maybe this is a little bigger here. Okay, that's wrong. That's completely wrong. The more PRB we have, the less tumor volume. Okay, so this is saying that when we have some PRB, we have an increase in tumor volume. Incorrect. Completely incorrect here. Okay, the more PRB we have, the more apoptosis. The more PRB we have, the less tumor volume. Answer C. I am very confident in my answer choice. Let's see if we got this right. Oh, I didn't put the review answer. Okay, let's see if we got this right. Bam! There we go. Let's keep going. Which statement is best supported by the data shown in figure 1? Hang on to every word in the question, guys. I will say it again and I'll say it a million times, okay? Hang on to every word. But Eric, it's only one sentence. Look, which statement is best supported? That means we're going to have some answer choices, okay? There's probably going to be an answer choice that's good, and there's probably going to be an answer choice that's even better. So we pick the better one, okay? So as soon as I see that, I'm thinking, I'm going to have some shit I got to go through, okay? So here we go. TNF alpha stimulates nuclear to mitochondrial translocation of PRB. Does it do that? Is there evidence of this in the passage? Okay, well, we know TNF alpha is pro-apoptotic, and we know in apoptosis, we involve that mitochondria. We involve a cascade of events there. Is it? Maybe. Let's keep going here, okay? PRB exerts its effects on TNF alpha stimulated apoptosis via the extrinsic apoptotic pathway. Here is a reach. This is very common in MCAT, okay? Here's a reach. They're reaching for the extrinsic versus intrinsic pathway. Have they ever mentioned intrinsic and extrinsic in this passage? No. So this is automatically wrong, okay? Whenever there's a huge reach, answer is more than likely wrong. Okay, here we go. TNF alpha stimulated apoptosis correlates negatively with induced PRB levels in back knockout cells. Okay, let's see. Remember, if something's negatively correlated, that means one thing goes up and one thing goes down. So let's see. Here we go. Back knockout. Here's a back knockout. Oh, almost dropped my mic. Hold on. All right. So here we have it. When we have basal to induced, this one goes down without TNF alpha. All right. Here we have with TNF alpha, we have basal, and then goes up. Okay. TNF alpha stimulated apoptosis correlates negatively with induced PRB levels. This is wrong. Okay. It does not correlate negatively. Okay. It correlates positively. All right. When we have induced PRB, again, what do we have? We have we have more apoptosis when we have induced PRB. Okay, we don't have less apoptosis when we have induced PRB. All right, this is how it is. If you want to get more specific on this question, guys, all right, This would be correct if they said backs minus/minus cells. Okay, the back is positively correlated. The backs is negatively correlated because look, it goes up, goes up, goes down. Okay, remember, we're always comparing the wild type. Therefore, this is wrong. All right, remember, if it said backs, then this would be right. But it says back, so that's wrong. PRB induction requires backs, but not back for TNF alpha stimulated apoptosis. All right, here we go. In this one, we have backs and we don't have back. In this one, we have back, but we don't have backs. So, what do we need more? Backs or back? Well, in this one, all right, when we have backs and no back, we have an increase. And this one, when we have back but no backs, we have a decrease. So that tells us right there that we need backs. Okay. If you want, I'll get into the figure right here, even further detail for you guys. Okay. When we have backs, we have an increase here, huge increase. When we're missing backs, we don't have much here. There's not much going on here. Okay. Remember, this is knockout of back, not backs. All right. It's kind of a little tricky there, but you guys can see it. Okay. And so we're left with A and D. Okay. Wow. It's 60 minutes. I'm going really in depth for you guys. This is an amazing freaking video. Make sure you guys drop a like here. Okay. So we have A and D here. Pick the better answer. The better answer is the one that's more supported by the figure. Write that down. The better answer is the one that's more supported. What's more supported here? The one that's literally supported in the figure. Okay, this, I can kind of make a reach here, maybe a little bit. This is directly right there supported. Fuck your reach. Reaches is a red flag. Therefore, we're going with D. Again, this could be good, but this is way better. That's the better answer. That's how you choose a better answer. Bam, D is right. Let's keep going, guys. Also, guys, if you haven't already, make sure you guys send me an email for tutoring and to join MCAT University, guys. This is the fully loaded program right here. It has everything, okay? And I make this very, very affordable for you guys because I know a lot of you guys do not have a lot of money, okay? So it's very, very affordable for you guys. I made it for every single pre-med. It's the fully loaded package, okay? You have 104 members here. You have other pre-meds to study with. They go ahead in this study chat. They always study with each other, guys. You're going to have schedule help, tutor notes, okay? You can ask me any question you guys have. All you got to do is go here, bam, and say, hey, Eric, what's this and that? I'll answer for you right there and then. You have help in every single subject, okay? Make sure you guys send me an email and say that you guys want to join. All right, it's $20 only a month. It's very, very good, guys. I wish I had this when I was, studying for the MCAT, all right? You really have access to me, a tutor. You can ask me questions anytime. Let's keep going. Which experimental approach would be least effective to determine the localization of PRB within a cell? Easy. Remember, hang on to every word. Hang on to every word. Here we go. Least effective. Fusion of a fluorescent tag to PRB. That's very, very effective. Very, very effective. If we fuse PRB with a fluorescent tag, all we got to look for is fluorescence. That's where it is. Wherever we see fluorescence, that's where PRB is. Very, very good. We're looking for the least one. Use of a fluorescent probe that hybridizes TRB transcripts. All right, if we want to find the location of PRB, we're looking for the location of a protein. This is going to tell us the transcripts, the RNA here, okay, that goes and, on the ribosome, gets translated, all that stuff. What good is it to tell us where the transcript is? There's millions of other transcripts there, okay? That's not going to help us at all. We want to know where it is. Just because it gets transcribed in the cytoplasm, or let's say it gets transcribed on the rough ER, that doesn't tell us much at all. That just tells us where it gets transcribed. We want to know where that protein is. So this is that one's pretty good. I like that one. Cellular fractionation to isolate different organelles. Cellular fractionation is when you get a solution of cells, you mash it up, grind it, whatever you got to do, run it into a centrifuge. Okay, and when you run it through a centrifuge at specific speeds, you're going to have I can't write on this, but you're basically going to have a different levels. Okay, when you run it through a centrifuge, the heaviest stuff is going to settle on the bottom, the lighter stuff is going to settle on top. Whatever's heavier is the one that has the most proteins. That one it's going to be your heavier, organelles, which is good. Okay, we're trying to find something. If we do a cellular fractionation, we can see, hey, is it in this organelle? Is it in this organelle? Obviously, we can't do an amazing job just with cellular fractionation, but it works. It works, okay? D, pull down of PRB and identification of interacting proteins by mass spectrometry. All right, guys. I am an MCAT tutor. Do I know what a pull down method is? Have I ever heard of that? No, I've never heard of it, and that's okay. They're giving us something that we've never heard of, okay? When they do this, it should raise a red flag. And also, identification of interactions, proteins. If we see what protein interacts with, we can trace it and find where that PRB is, okay? It's that simple. That simple. We know PRB, let's say, interacts with back or backs. We find backs and back and look when it interacts. That's a great way. This is a very good way. And so we have B and C. Again, this is a good way, and this way sucks. So answer is B. That is a better answer. Bam! There we go. Let's keep going. Let's keep going, guys. I'm having fun here. You having fun? I'm having fun. Here we go. Rhodamine 123 is a fluorescent dye that binds to polarized membranes and is used to label mitochondria. A cell line expressing basal levels of PRB, or which form of induced PRB would most likely exhibit the lowest level of rhodamine staining. So rhodamine, our guy rhodamine here, binds to polarized membranes and is used to label mitochondria. All right. And they're asking RBN, PRB, RB. Okay. Let's look at here. All right. Think about it, guys. This is a pretty good question. It requires you to think, okay? If we have more apoptosis, is that mitochondria more likely to be there? More apoptosis means less mitochondria. Why does it mean that? Well, because in the mitochondria, we have the whole entire cascade going on, okay? So, the more apoptosis, the less mitochondria. The more apoptosis, the less labeling. Why? Because rhodamine is used to label mitochondria. If we have less mitochondria, we have less labeling. Less mitochondria equals more apoptosis. That is AMC logic again, right there and then, guys. Right there and then. That's how they want you to think, guys. You think like that, you'll get all these questions right. It's this goes up, this goes down. Blah, 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 blah. That's it. That's literally it. Well, for the BB and CP. I don't know about freaking cars, okay? Cars is another nightmare. So we just look for the one that has the most apoptosis, okay? Here we go. PRB induced, good amount. RB underscore N, down here. RB underscore SP, up here. More than PRB induced. RB underscore C, down here. All right. So automatically, guys, the answer is the one with the most apoptosis is RB underscore SP. Now, here's another trick I have for you guys. And this video is absolutely amazing. This is so good, this video, okay? Look, RBN and RBC are the same exact thing. Whenever you have an answer choice with two answer choices of the same exact thing that mean the same thing, they're both wrong. Boom. Wrong, wrong, automatically. I promise you guys 100% of the time. Always, 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 okay? It's common test-taking strategy there. So I'm pretty confident, C. And that's how we do. We're confident as fuck in all our answer choices. And that's how we don't, that's how if we're confident as fuck, then we're not going to go ahead and overthink and say, hey, maybe it's this, maybe it's this. No, we're confident as fuck. That's what it is. We keep going. Excuse my cursing. Oh, no. Oh, my God. I spoiled this one. Okay. Let's, all right, here we go. So the caspase activator released during the mitochondrial autophagy pathway primarily functions in which cellular process? Okay, this is pretty simple, guys. All right. How do I freaking go back? I don't want to do this. All right, whatever. All right, I guess we're going to do this together. All right, so it says caspase activator that is going on cytochrome C. Okay, if it's in cytochrome C, where is cytochrome C? Cytochrome C is in the electron transport chain. All right. Cytochrome C does not involve proton pumping. Which parts of the electron transport chain, which enzymes pump protons in the intermembrane space? Which ones? It's complex 1, complex 2. No, not complex 2. It's complex 1, complex 3, and complex 4. Only ones that do that proton pumping. Cytochrome C is just going to transfer electrons. That's why the answer is D here. How do I don't want to do this. I don't want to choose the next answer choice and then it's going to show the answer. So I'm going to do something real quick. I'm going to reset the question, then go back to it. Okay, so hold on. So we're done practicing. How do I... all right, here, reset all questions. All right, we're gonna go here again. All right, we're gonna go back. We're going back. Excuse the technical difficulties, guys. All right, here we go. All right, cool. So we did that one. That was electron transport. That was pretty easy. Content review there. Okay, here we go. Last question. Cells were incubated with the indicated amounts of recombinant PRB only or recombinant PRB in the presence of backs and back. Following centrifugation, cell membranes were disrupted, but organelles remained intact. Levels of cytochrome C in the supernatant and pellet fractions were analyzed by Western blot. Which graphic shows the expected result? All right, whenever you guys see answer choices with these Western blots here, plus, minuses, concentration, another one here we're looking at, another thing here we're looking at. Don't freak out. I know a lot of you guys freak out. It's pretty simple when you guys think about it, okay? Because it's either this or that. It's that simple, all right? For this question, I'm not going to lie, this is a pretty heavily worded, difficult question, okay? So if you get this right, if you get this wrong, don't feel too bad for yourself because this is already a kind of like a bullshit question, but we're going to do this right now. We're going to get it right, okay? So, cell membranes were disrupted, but organelles remain intact. All right, again, centrifugation, guys, is when you go ahead. All right, I'm going to bring it up for you guys so you guys can see. Centrifugation. All right, here it is. All right, you put this in the spinning thing, and due to, centripetal force, all that stuff, it's going to spin, spin, spin. You're going to have the pellet here and the supernatant up here. The pellet here is what's heavier. This is what's lighter. That's simple, okay? Now, they told us that the cell membranes were disrupted, but organelles remain intact. So the organelles are going to go on the bottom, and anything that's not in organelles is going to go on top, all right? So here we go. And then levels of cytochrome C in the supernatant pellet fractions were analyzed by Western blot. All right, cool. We want to see where levels of cytochrome C are. Are we going to have cytochrome C in the cytoplasm, or are we going to have cytochrome C in the pellet? Okay, the supernatant, we can consider it as the cytoplasm because literally everything else besides the organelles. And what's everything else besides the organelles? Cytoplasm, okay? So here we go. Again, if these don't get scared, guys, let's go take it one at a time here, okay? Here's PRB, 0, 500, backs and back, with backs and back, without backs and back. Here's cytochrome C. Do we have it in supernatant? Do we have it in the pellet? Let's see, okay? So, if we have no PRB and we have backs and back, A is telling me that we're going to have no cytochrome C in the supernatant. All right. If we have apoptosis and a lot of apoptosis, the cytochrome C is going to be in the supernatant. Okay, I'm going to say that again. If we have a lot of apoptosis, all right, the materials, what's in the mitochondria, is going to be disrupted and it's going to be released in the cytoplasm. All right. If we don't have apoptosis, then the cytochrome C, which is material of the mitochondria, it's going to remain in there. It's not going to get exploded or anything. It's going to remain in there. And that mitochondria with the cytochrome C is going to go ahead and be in the pellet because it's intact. All right. If we have blown up mitochondria, cytochrome C will make its way to the cytoplasm, aka the supernatant. If we don't have apoptosis, we don't have that explosion of mitochondria, we don't have that explosion of the mitochondrial proteins inside there, we don't have that explosion of cytochrome C, then cytochrome C is going to remain in the mitochondria and settle in the bottom of the centrifuge tube and be in the pellet. That's what it is. All right. So PRB 0, backs and back. Are we going to have apoptosis if we have PRB 0? We're going to have very, very little, maybe none. Okay. When we induce PRB, we have a lot of apoptosis. We saw that here. Okay. So, and if we induce apoptosis, we're going to have that cytochrome C in the supernatant in the cytoplasm. If we don't have apoptosis, it's not going to be in the supernatant. It's going to be in the pellet. Okay. Makes complete sense here. Complete sense. All right. Let's look at the third row here. 0, PRB, no backs and back, and no PRB. Boy, we're not going to have no apoptosis there. None. We need backs and back. They involve the activation of apoptosis. We're going to have no apoptosis if we don't have backs and back. Therefore, no apoptosis. All that cytochrome C is going to stay in that mitochondria and not be exploded out. I like A a lot. And if we want, we could do the fourth row. If we have PRB, this is going to induce apoptosis, but we don't have backs and back, bro. We need backs and back for the activation of apoptosis, okay? And that's why you see barely anything here. And that makes sense. No backs and back, no activation of apoptosis. All that cytochrome C is going to stay in the pellet. Bam. I like A a lot. And if you compare A to B, C, and D, then I'm just going to repeat the same things, but I'll do one here just in case. If you understand it, then you're good. You know, subscribe, click out the video. But if you don't really understand it that much, I'll explain it one more. I'll compare it to C here. Okay. So again, guys, if we have apoptosis, that cytochrome C is going to move from the mitochondria, be exploded out into the cytoplasm, aka the supernatant. So what does this say? PRB 0. All right, we have little to no apoptosis with backs and back. Again, if we have little to no apoptosis, okay, that cytochrome C is not going to be in the supernatant. That cytochrome C is going to remain in the mitochondria. C is telling me that cytochrome C was exploded out of the mitochondria and went to the supernatant / cytoplasm, which is wrong. Okay. It's not going to be exploded out because we don't have induction of PRB. So C is wrong. And you can do the same things for B, C, and D. So A is right. Oh. Bam. All right. That's it for this video, guys. Make sure you guys go ahead and sign up for MCAT University, guys. It's very affordable for you guys. It has everything that you need. It's loaded up, okay? You guys can study with other pre-meds. You can ask me any questions you have in any type of your content review. We have help in every single subject, guys. I'm there for you guys. You know, if you could ask me, hey, what should I do now? What should I do in the last week of my MCAT studying? All that. And we have help in the passages as well. It's a fully loaded program. It's $20 a month only. It's very, very cheap. Very, very good. Okay, so send me email at erickmedicine1919@gmail.com if you guys are interested in joining. That's it for this video. I'll see you guys in the next video. Peace.